There's 44 slides, but a lot of these are things we can skip, so we should probably like 30 or 25 slides. There's a calculation at the end, and there is a puzzle problem in the middle. So there's a few important things in here. The whole idea of this chapter is to start using what we now do. DNA sequence. What can we do from sequencing microorganisms and also people? What types of things do we notice? And when it comes to humans, we typically use this term polymorphisms to describe basic differences between one individual and another that don't necessarily have an associated phenotype. A lot of polymorphisms tend to be neutral, really no effect, either bad or good. There are some that are really bad, and we'll talk about those as we go along. With DNA sequencing comes genetic analysis, and that leads to a lot of phylogenetic studies, or molecular studies, or ancestral studies to compare one person to another person to see are they related, are they not related, some really important things. And if you haven't taken 391 yet, you might pay attention to the allele frequency calculation, because that can definitely help in 391. It'll also help for later when we do something that's not quite Hardy-Weinberg, but beginning. Which is fun. If you know what I'm talking about, you probably dread me saying that. But it's not Hardy-Weinberg. This slide I've shown you before, but it is an example of what we can do with the power of DNA sequencing and computers. Really, without having really good computers, DNA sequence analysis would be caveman type of things. Because even though there's only 4 nucleotides, if you're looking at 1 million nucleotides for 50 different people, you got to be able to line them all up to make sure they line up the right place, and then you got to see are the differences and what they are. And again, if we compare the genomes of different individuals, we can see that there are differences. And some of these differences are ones we'll talk about. Some of them we'll pick up a little bit later when we get into meiosis. The DNA sequence is the wave of the future, and genetic analysis, genetic profiling, genetic screening. You've never heard of any of these terms. That is the thing. Whether it's breast cancer, colon cancer, trying to figure out what kind of dog you have. It's a thing. There's a company that will tell you what kind of dog you have. You don't know. So there's a lot of power in DNA sequencing, especially when it gets to dogs. Certain dog breeds are prone to certain types of disorders more than others. So knowing that your dog is part one dog might help you identify whether or not hip dysplasia or whatever is going to be a problem with that particular breed. But a lot of times when we look at DNA, we don't have a phenotype. So we can't say you're blue and you're red. It doesn't work out. It's just you're A and you're C. That might not actually have a phenotype. So we're going beyond epistasis ideas and penetrance ideas and pleiotropy ideas. Just look at the DNA sequence and compare one individual to another individual. And based on all the sequencing we've done of humans, there's only one change every 1,000 base pairs. So look at the person to your left and the person on the right, and there's only 1 in 1,000 difference between you and the person sitting next to you and around you. Does that seem low or does it seem high? We have a lot of different varieties of people in here. So I think there's only not that many, 1 in 1,000 differences between you and the person next to you. It's pretty remarkable. There's a lot of these things that are out there. Actually, it's pretty often. 1 in 1,000 is not that, it's pretty low. It's like a fairly often type of thing. But we still have what we have. Again, a lot of these are not really related to anything. We call them polymorphisms. And a lot of them will happen in genes, but also a lot of them will also happen outside of genes. We have got these things that we can deal with. Now, why do we care about DNA sequencing? Because genetic testing in humans does not work the same way that it works with other plants or animals, or actually plants or other animals. With a plant, you can control where does the sperm and egg come from. You can make true breeding plants. Imagine trying to make a true breeding human. What would that look like? What would it entail to do a true breeding human? It'd be very hard to imagine. Might only work in one group. Because women can have children and men can't have children. So that kind of complicates things. Back crosses, test crosses, reciprocal crosses, hard to do with humans. Because People don't want to be told who to breed with and how often to breed and how many children to have. But what we call brood size, that people don't think of humans as brood size, how many children do you have, is really low. 1 or 2 children per couple, sometimes 8. Depending on fertility tests and all. We have very few children, and so there's not a whole lot of statistical analysis that can be done with human genetics, doing the basic phenotype analysis. So that's where DNA sequencing plays a very crucial role in this process. Eliminates all the need for doing testing that was Gregor Mendel and all of that early genetic analysis. Just look at DNA sequence. A lot of people are trying to do some really cool things with DNA. I found this paper, it was earlier this year in February, but it was a story about a paper trying to determine if there was a genetic component to fitness. What kind of things can we find in our DNA, and what does that tell us about your ability to jump really high, or run really fast, or shoot a ball really well, or to hit a ball really well, or kick a ball really well. This is in our DNA somewhere. You're inherently born with a certain innate ability. So that's a genetic component. Like, you don't learn how to kick a ball until you learn how to kick a ball. You're either really good at it or you're not really good at it. So, is there a genetic component to being the ideal athlete? Maybe there is. But the worry about all this genetic analysis is what kind of mad scientist is going to come around and decide that only certain people can have children and only certain children can be born. Which is, again, the big ethical, moral problem with being able to do genetic engineering is what should we do, not what can we do. The 3 most common types of polymorphisms that we will talk about are what are called, first, the SNPs, SNP, called SNP. They are the most common and the smallest, and they're going to be not really that dangerous. We have SNPs as one of our type of polymorphisms. The next is called the STRs, the short tandem repeats. If you were paying attention on hunting, you might remember the Y chromosome has a lot of these STRs present on it. The last is called CNV, for copy number variation. And as we go along, we will talk about size, abundance, and detriment. So how bad are they types of things with each of these. And basically, we start at the top of not bad, really small, to really big, really bad. And SDRs are in the middle. But as the name implies, a single nucleotide polymorphism is a single nucleotide change between one individual and another individual. And what's cool about the SNPs is you have lots of different types of phenotype or genotype varieties. Because you could be homozygous or heterozygous or opposite heterozygous, depending on how you think about things. Or it could be other homozygous. And so each of us has 2 copies of the chromosome, and we're diploid organisms. So maternal, paternal, you could have the exact same nucleotides in both, or they could be the same but different, or they could be different. And also this could be reversed. This could be in the maternal, and that could be in the paternal. And that could change things. But these are the things we think about with single nucleotide polymorphisms. The single nucleotide changes. How bad is one nucleotide change? Anybody have any thoughts? How bad is one nucleotide change?
Depends on where it is.
It does change. It does depend on where it is. It also depends on what it is. There are 2 really important things that will define the severity of a single nucleotide polymorphism. Single nucleotide polymorphism. But generally speaking, it's 1 nucleotide, so it's really small. Not a whole lot of DNA is affected. The effect is going to be variable, because we don't really know exactly what it's going to do. But chances are this is going to have the minimal effect. As we think about these 3 things, this is the one that is least likely to cause a very detrimental and deleterious effect. Because it is only 1 nucleotide, and it's really not that important sometimes. In sequencing chromosomes, we have seen some trends, and one trend to be aware of is that SNPs are more common in non-coding regions and less common in coding regions. Is this chance, or is there a design behind this phenomenon? Does anybody have any thoughts on why coding region mutations would be less likely to occur, or less often observed? What's special about coding regions? What does coding region mean?
They're expressed.
And what does expression mean?
They're seen in the phenotype.
Go back a step further. Before we get to phenotype, we have to make something. Why don't you just think about what does gene expression do?
Make a protein.
Right, so different gene expression makes a protein. Coding regions mean protein coding regions. And if you change the DNA of a gene, you have the likelihood of changing, at least the potential, to change the amino acid sequence of a protein, which could affect its function. If these mutations happen in the non-coding regions, who cares? They don't make proteins, so these things are more allowed here and less allowed here, evolutionarily speaking, because changes here might make you die quicker and not reproduce. It might not be seen and carried over to the next generation. Whereas these might be carried over to the next generation more often or more quickly. And these are the majority of those differences that we see. Because there's one every 1,000 or one every 2,000, or an average one every 1,000. And just for thinking about 3 billion base pairs, which is not the size of a human. Actually, no. Yeah. That is the genome size of humans. You could have between 1.5 to 3 million of these things. That sounds like a big number. But we're all still 99.9% identical to each other. Every human is 99.9% identical to each other. That 0.1% is the differences that we see. So not a whole lot of variety in humans. You know, we do see different skin colors, eye colors, hair colors, height, weights, what have you. On the genetic level, there's not a whole lot of difference. And we also have to think about half of the genome is transposons, stuff that doesn't really matter. So maybe there aren't as many of these changes in there as we might think. Because it is a single nucleotide, the only way to identify an SNP is through the process of DNA sequencing. There are other ways to test things that are, identify things that are bigger, like for STRs and CVs, but the only way to identify a single change is through DNA sequencing. Now we get a lot of these things. Again, everybody is going to have its own little SNP sequence or pattern that is unique to you. If we sequence everybody, we'd be able to identify everybody pretty quickly. No longer do we need dental records or fingerprints. Maybe eventually you'll have a DNA sequence. Imagine if you walk around with a card that has a piece of your blood on it. And that piece of blood goes into a machine and reads your DNA really quickly. That's like what science fiction says. You can just sequence in like 3 hours. It takes longer than that. A lot of these don't have any effects. So they're there, but we can't measure them, so that's why DNA sequencing is useful. A lot of people are trying to look at the importance of SNPs. A lot of genetic studies are trying to identify if there is some kind of true genetic component to certain types of diseases, and this paper is based off of looking at certain types of cancer. So by sequencing a bunch of people with cancer, can they identify a sequence component that is indicative of a risk for a certain type of cancer? And we talk about this a lot. Genetic screening for colon cancer, breast cancer are pretty common. But it's lots of different types of things that we can try to screen for. Now this one is kind of fun. Has anyone ever seen their doppelganger before? Does anybody know what the doppelganger is? Someone that looks like you, but it's not you. Now if you have a twin, it doesn't count. Doppelganger is a genetically unrelated individual that looks like you. And a group of scientists got together and tried to identify or compare, genetically speaking, doppelgangers, people that look alike, but aren't genetically related to each other. What can we see? And there's a lot of things. They sequenced every single chromosome to figure out what was going on. And just as an example, if you look at chromosome 2, we see certain types of changes are more common than other types of changes. Does anybody remember anything special at A and G and T and C? A and G have a certain name because of their shape. Does anybody remember what that was or is? A and G are purines. What's T and C? Called pyrimidines. So if you look at the list, a purine-purine change or a pyrimidine-pyrimidine change is the most common difference between an individual. The other changes, which are purine to pyrimidine or pyrimidine to purine, less often, and then some of them are very not common at all. And this was true in every single chromosome. They found this type of trend. And the basic idea that I want you to think about is some mutations are more common than others. And there's a word for this. It's not on the next slide. Unfortunately, it's on a slide later on. But because we're here and because I like to preview things for later on, this word will not show up on exam 1, but should be on exam 2. There are two types of changes that we typically see. The first is called a transition, the other is called a transversion. And these two speak to what type of change do we see. Transition is purine to purine or pyrimidine to pyrimidine. That's the most common change we see, a transition type of mutation. This is anything else that's less common. And typically this is about 2 times more common than the other type. So when we talk about SNPs kind of being random, they're not really random. There is an order to the nucleotide changes that we typically see. There's an underlying reason for these changes, which will take us to talking about the genetic code. Do you remember anything about the genetic code from 140? Tells you which 3 letters which equal which amino acid. Vaguely, something like that, somewhat. I think it's really interesting that sometimes they detect none of these changes. Now this paper looked at every single chromosome, and I'm going to show you one. But some mutations are more common than others. I'm not talking about coding versus non-coding. We're talking about actual nucleotide changes. So we'll revisit this very important idea about transitions and transversions. Again, that's an exam 2 thing, not an exam 1 thing. The next type of change we see is the STRs. We're going to get bigger. The word repeat is important. That means several nucleotides repeated over and over again. So just by sheer number, this is always going to be bigger. I don't really know what to say here. We'll say, large doesn't really seem like the right word. Large seems too big. Big seems too big. Large-ish. It's not small. Some of them can't get very big. But as we're going to see for C and Ds, we're going to see very big. So we definitely see a difference in the amount of DNA that's affected by each of the 3 different polymorphisms. Like the SNPs, a lot of times the STRs have a minimal effect. But that effect can be bad. And what really is important with STRs is the number. How many actual repeats do you have? This is also generally individual. The number of repeats you have in a particular location could be new or different than someone next to you. But they can also be used to study ancestry. Certain groups of people might more often have a high number of repeats of one and a low number of repeats of the other, or reverse. So these can be used for ancestral analysis. And I would say what a lot of what 23andMe and Ancestry.com is STR analysis. They're not going to see what's your entire genome. They're going to see what's parts. And they've identified parts that are more or less useful in identifying lineage or inheritance patterns or based off of, different types of people from different parts of the world. And so just to give you an idea, these are a little bit more common than the S&Ps, but 1 in 2,000, really not that different between 1 and 1,000. Some of the S&Ps were 1 in 2,000 also. So they're relatively pretty common as far as what we look at as commonality speaking. That's one, not on the board. How common is this? Some of them are pretty long, some of them are more than 100, sometimes even 1,000 repeats that we can find in places. If they happen inside a gene, that can have a pretty big effect. If it happens outside a gene, maybe it's not so bad. The SDRs are really important. This is the paper, it's an old paper, but it looks at SDRs and what you can do with them. But in this paper, they're specifically looking to see if any of these SDRs were associated with any human diseases. And there is one that we'll talk about, a very important SDR that is affected by repeat number. But it's not in this set of slides. It's in the next set of slides because it relates to repair. And it's not about just looking at the things that are there, which is what these slides are about. What is there? What type of things can we see? You can identify this by sequencing. Everything can be identified by sequencing. But because of the number of repeats making a size difference, we can use gel electrophoresis as a way to identify size differences. To get the DNA to analyze by size, we need to do something called PCR or digest DNA. We'll talk about PCR as a tool when we get to talking about replication. If you don't know anything about PCR, it's a way we can make a little bitty piece of DNA and analyze that small piece of DNA. But like SNPs, SDRs are pretty common. They typically don't have a lot of phenotypes associated with them. But they can be used for doing various things. And this is where our problem-solving skills are going to come into play, what we call SCR analysis. And so we can look at one individual versus another to see, is there a genetic relationship between parent and offspring, or offspring and parent? I don't want to go all Maury Povich on everybody. Raise your hand if you know who Maury Povich is. Well, Jerry Springer. You may know Jerry Springer? One of the more common shows that's on Maury Povich and Jerry Springer is Who's My Daddy? You ever watch those shows where somebody says that this guy is my father, my child, and I want you to test him? It's a common test. It's a paternity test. It's done a lot. But a lot of it is just based off of STR analysis. And so you PCR up certain regions of the genome and then run that DNA on a gel. So for DNA gel electrophoresis, small pieces go where? Where should the small pieces be? They go further, which makes them near the bottom of the gel. And typically when we look at gels, they're oriented, this is the top, this is the bottom, so DNA is going to run in this direction. And smaller pieces will always be near the bottom, bigger pieces will be near the top. So we can separate things by size. And I just want you to focus in on one group. We'll focus on these three right here, because that's parent and one child. Diploid organisms, so you can either be homozygous or heterozygous. And so for this individual here, the fact that there are two pieces of DNA indicates that that's heterozygous. So on your maternal piece, the STR is this long, on your paternal it's this long, or it's reversed. For the other parent, they get sizes that are down here. So different numbers of repeats in mom versus different number of repeats in dad. Now remember, when sex reproduction occurs, a portion of your DNA comes from one parent, the other portion comes from your other parent. So you're going to inherit something from mom and inherit something from dad. So our child is in the middle, doesn't matter if it's male or female. But the fact that both the mom and the child have this piece of DNA in common, says that this piece of DNA was inherited from mom and not from dad because dad didn't have that piece. This piece of DNA here was inherited from dad because you see they're at the same size. And so just by flip of a coin, you didn't get this one, but you got that one. You got that one, but didn't get that one. That's genetics. Flip of a coin, 50-50. You get mom or do you get dad? The same thing is true for over here. You see, we look at mom, dad, and the child. Both mom and dad have the same SDR. So you can't really tell anything about, did this come from mom or did it come from dad? So look at the other one. And based off of this, did this come from mom or dad? Square is father, circle is mom. So did this come from mom or dad? It's the same size as this, right? So it has to be from this pair, which is dad, which means that came from your mother. So again, flip of a coin, you either get this one or that one, or you get this one and that one. One of these only has one, no, none of these have one band, this one sucks. On some of them, only one band appears, which means you're a homozygous. But in all these cases here, they're heterozygous. But again, if you were to look through these 7 children right through here in the middle, can you identify, are they from this side of the family or from that side of the family, genetically speaking? So look at the one here in the middle. Is this a mom's side or father's side thing? So the father's right here, mom's right here. Yes, just looking for where do these things overlap. This is kind of confusing because of the way it's organized, but where do we see this in our parents? It's not over here. It's definitely over there, so that makes it a paternal thing. And then this one down here is only present in your mom, so it's a maternal thing. So we're just looking at size comparisons. Just draw obviously a circle around something to see, is it in the same position as something else? This is one of the pictures from the textbook, and it's the same type of idea, but it's an actual picture of a genetic test done in real life. C is for child, M is for mother, A is for alleged father. And then we can ignore the A plus C because that really doesn't matter. But in our child, look at what DNA is present. There's this band here and then that band there. So again, we only see 2 bands because we're looking at just one little thing. And so trying to decide, is this a potential father of this child? Would you say yes or would you say no? There's a chance because both the father and the child have a DNA piece in common. The other piece you can see came from mom. And mom's other piece is not present in, that is weird, because both mom and dad have that same piece of DNA down there. This is where we see the difference between mom and dad. Here's another example. And compare child to alleged father. Is this alleged father potentially the father of this child? What do you think? Who thinks yes? Who thinks no? Who says, I have no idea what's going on for children? Okay, we're just trying to look at comparing things. And it might be kind of hard where you are, but if you look down at mom, or compare mom to child, the mom doesn't have a band over here, but the child does. The father also doesn't have that band. So the fact that both the mom and the dad don't have the DNA in the child suggests that it can't be from that father. Because it doesn't come from mom, it has to come from dad. So that indicator is a pretty important thing. Here, child doesn't have that band that's in dad, and child doesn't have that band in dad either. So this is not the dad, yay, or sad face, depending on how you felt about being the father of this child. But we can see in between mom and child, there is a common band between those 2 child and offspring. But for dad and child, that's not the case. For the exam next Wednesday, you're gonna see a picture that looks something like this. And what you should be able to do is tell me who are the parents of child E, who are the parents of child F, G, child G, H, and I. So which two parents, between mom and dad, which two will be the parents of G, H, and I? Go ahead and take a second. Do it at home at your bench. And we'll see if we get the same answer or if we get all the different answers. I'm trying to compare. So look at the child and think which adult has that same piece of DNA. And you match up one mom, one dad with each child. On the test, when I do it, there'll be lines going through things, so it'll be very easy to distinguish sizes because they'll all be on a separate line. But for child G, which dad? That's this symbol here, if you didn't know. So we're looking for, go across, we find, there's one, which is most likely the right one. No, that one could be. Yeah, look at this one, there's only one. So dad is E, then mom, most likely is C. What about for H? Who are the parents of H? And all these are the same size. So it looks like this one mom is the mom of all three of these children. The dad is not the same. So who's the dad of H and I? Because H and I have basically the same piece of DNA here. So H and I are related to each other beyond just being from the same mother. And just, go across, and E is the guy. So here we have a case where the children of this woman have two different fathers. I know. But different piece of DNA inherited from each individual. So yes, E and C are both the parents of G, H, and I, but genetically speaking, they are not all the same. Which is what you expect. Children, can be different. Somewhat dad, somewhat mom. Sometimes things skip generations. We do see some weird things. But there should be a picture or a couple of these little pictures on the reviews for you to look at the practice. With the copy number variation, we get back to the idea of a gene dosage as a problem. Remember, gene doses is something that's going to make you significantly different than other humans. Now with copy number variation, we are going to see the largest amount of DNA affected, which is also going to give us the worst type of possible effect. Because there's lots of changes, and these changes typically have some really deleterious effects. The size is not important, but compare one nucleotide to 300,000. It's a pretty big difference, right? Of how many pieces of DNA or how much DNA is affected. You might have, millions of SNPs, but you don't have very many of these. These are the rarest cases of things. Most of you probably don't have any, which is good because you're hopefully mostly regular. Some of you might have already died already. And it's kind of flippant, but there is one case of cognitive variation that does result in death of adolescents. What is so bad about copy number variation is you do get extras or less. It's not exactly the same thing as a true monosomy or a true trisomy. If you copy half of a chromosome, you have an extra bit that no one else has. And that's the order of magnitude we are, like a half or maybe a quarter of a chromosome might be duplicated. And that's 25% of the monosomy or 25% of the trisomy. So it's a pretty big effect. And what typically is going to lead to copy number variation is what we call a duplication or a deletion. Something that'll also be more important to remember for exam 2 and less important to remember for exam 1. Because when we get to talking about mutations and things, we'll go revisit SNPs, STRs, and copy number variants. To remind you, this is the thing that went wrong to make this happen. So if you have 2 copies of a gene when you should only have 1, you got extra something. Or now you have 0 something, or less overall. And just as a textbook use thing, this study was done to look at the genetic component of people from regions where different diets are used. I don't know if you noticed anything about some of these populations. Some of these populations are very heavily rice-based. That would be sort of Japan area. But rice is a starch, and what this study looked at was how many copies of amylase do these individuals have in these regions. And what they found was a correlation. Populations that ate more starch tended to have more copies of amylase than populations that didn't eat as much starch. So is this the effect of diet affecting genetics, or is the effect of genetics affecting diet? Jane and Ed, what do you think happened first? The diet or the genetic change?
Diet.
I would probably agree that diet is probably here and having an effect on genetics. Because why change? Or why be different? And in some of these populations, they're really close to each other. And so it seems really weird to have a close proximity be very different in genetic makeup, but here, diet probably has an effect on how many of these things you eat. It's not just whether it's starch, but a lot of diseases are affected by these, and they're called, what does CHD stand for? Congenital heart defects. Has anybody ever heard of somebody just dropping dead at a football practice? That, in a lot of cases, is tied to this type of CNV. And one that was in the paper recently, and by this I mean probably 5 or 6 years ago now. But they sequenced the genome to figure out where it was. And not that you need to memorize any of this, but if you care, it's called studying sudden cardiac failure. And this particular individual is because of a duplication of not just the gene, but a lot of DNA around the gene. So you just get this extra thing there that has a problem. Now the reason they want to do this is because they can trace this back to phenotype. If you have this extra bit of this DNA, it makes a non-functional protein. So now you have non-functional and functional of these protein, which are important in muscles working or not working. So you can try to see, what happened here. And then it is a very rare thing to happen. Sometimes you might go through your entire life and not have to worry about whatever would happen here. And I like this term down at the bottom because it's fun. Highly penetrant. What does that mean? Highly penetrant. So this is the definition of penetrance, and try to put it into context here.
Very often expressed when it's present.
I think you're on the right track. I just wouldn't phrase it that way. It's not about spread. But penetrance is how often does the genotype make the phenotype happen. And so if you're highly penetrant, then it's 100% chance that phenotype is going to be there with your genotype. So keep in mind, some diseases have very low penetrance, others have very high penetrance, meaning your likelihood of getting it is pretty high if you have the right genetic makeup for it to happen. So everything is about DNA, and we can look at RNA as well. Everybody likes to use the word omic for something, because that was the big buzzword for a while. Let's look at genomics. Compare the genome of one individual or another, or one animal and another animal, or bacterium to another bacterium. Let's look at proteomics. What proteins does one organism have versus what proteins does another organism have? Let's look at transcriptomics. What RNAs does one have versus what RNAs does another have? And then there's fun things like metabolomics, which is trying to use DNA to piece together what types of chemical reactions go on in an environment. All things that will not help you on a quiz or exam, as far as those definitions go. But people make some weird words, and you can make a weird word for whatever word you want to make a weird word about. But it's all about comparing things, and trying to piece together a puzzle. And sometimes the puzzle is, what chemical reactions can we predict are happening based on what DNA sequences we have detected in a region? Or what proteins have we detected in a region might tell us what enzymes are there, therefore what's going on in a system. When it comes to microbiology, it can't grow everything in the lab, so these types of studies help us study the things that we can't grow in the lab at home. And when it comes to omics, one of the more important omics is comparative genomics, where we're trying to compare the genomes of different individuals, and ancestry is one of the major things we care about with comparative genomics. If you've never seen this picture before, it's basically the mitochondrial DNA movement from where the first human was to where all the humans now are. And you basically say that this is where the first human was, and then that one made offspring that went out and populated the rest of the world. That's what evolution and comparative genomics tell us. Again, this is about mitochondrial DNA, because we're looking at the maternal relationship of parent and offspring with each other. What is more important about the slide is these 2 points here. And this is directly related back to the idea of S&P frequency. Where are S&Ps more likely to occur? Coding or non-coding areas? Non-coding, remember non-coding, we don't care about it because it's not going to change our protein functions. And so coding regions are slower to change because S&Ps are sort of acted against, selected against might be the best word also. There's an evolutionary drive to keep your genes the same, keep your proteins the same, to keep your organism the same. But the non-coding regions can change. So things that stay the same, relatively speaking, tell us how things are related to each other. Things that change very quickly tell us how far apart things are. So those non-coding SNPs are pretty important because they tell us divergence, or this idea of how different are we relative to anybody else. The similarity versus differences are the two types of things we look at. And coding regions and non-coding regions tell us one or another. It's not exactly true for what Carl Woese did when he looked at the DNA for the ribosome, but in the ribosomal RNA, there are regions that change very quickly and regions that change very slowly. So you have places for comparison, say they're similar, but also places where they're different, say that they're different. And just to give you another fun little picture of using molecular genetics or comparative genomics to study people, we have probably all the people on the planet, relatively speaking. Although there is a year difference here, so these are early humans, these are sort of where we are right now. So if you just look at this top part, does anything up here kind of strike you as weird? I don't know if you can read from where you are, but Italian is down here, German's right there, French, Dutch, English, Korean, Chinese. Chinese is also up here, and Japanese. Why isn't Germany next to Italy? I guess they're really not that connected to each other, right? But is it weird that most of the European nations are not really right next to each other? Is it weird that some of the African nations are not really represented here either, and they're in things they don't really know? Yeah, something to look at. It's this one. I can't remember which one it is. It's one of these. Or maybe some of these up here. I can't remember. There we are. Why are there two Australians? One up here and one down here. What do you think? Sir, what? I think it's an Aboriginal versus a non-Aboriginal difference. So you have Native Australians and you have the people that came from England to populate Australia. These are some fun things we can do with genetic analysis. You can see peoples and how they're connected together. I know I just have a few more minutes, but squeeze it in, squeeze it in, squeeze it in, squeeze it in, squeeze it in. Yes, yes, thank you. Squeeze it in. Here we go. Has anyone ever thought which are related most to? Chimpanzees, gorillas, orangutans, or baboons? Don't call anybody a baboon. It's not that true. Call them a chimpanzee. Pretty true. Chimpanzees are our closest relative. That's a non-human relative. And gorillas are next, and then orangutans, and then baboons. Does anybody know the exact difference between humans and chimpanzees? Look ahead one slide if you don't care. Cheat. There's a chromosome difference. We have 23 pairs of chromosomes. Chimpanzees, gorillas, and other primates have 24 pairs of chromosomes. And so in doing the genetic analysis of a human genome versus a chimpanzee genome, here's where the differences lie. Chromosome 2. In humans, chromosome 2 is one piece. In chimpanzees, there are 2 chromosomes instead of 1 chromosome. So evolutionarily speaking, scientists had to decide what happened. Did this one break to make these 2, or did these 2 combine together to make this one? And how do you look at this? What's at the end of the chromosomes? What we call this piece of DNA is located at the end of the chromosome.
Telomeres.
So telomeres, lining things up. What do we have here in the somewhat middle of the chromosome? A centromere. So those are identifying characteristics. Repeat sequences of the telomeres, repeat sequences of the centromere. And so lining up them, this one lines up here, and you have a nice centromere there. But what about here? There's a centromere right there in that one. And what they found by analyzing chromosome 2, there are remnants of a centromere right here. And so these 2 things combine together to make us. So I asked you earlier about, does chromosome number matter? In this case it does. Because we have almost exactly the same genome as another animal, but the actual number of chromosomes has changed. There might be a few other variations, but you can see some of them was lost. You just compare these bands, that piece is there, those pieces are there. What happened here? You sort of just lost those telomeres and then glued them together in some way or another. So, this is kind of a fun little story. Eventually you'll be asked to remember this story. It's not a breaking process, it's a fusion process.